Good morning, everyone.
Good morning.
Okay, welcome back.
Good morning.
Good morning. Welcome back, everyone. I hope you're doing well. I know 8 a.m. classes are always difficult. So thanks for being here this morning. Okay, well, folks, that time is upon us. What I mean by that is we're getting ready for our first lecture exam, right? You have lecture exam one. You have lecture exam one this coming Tuesday, October 12th. So let's talk a little bit about that. Let me just remind you of some things. And I'm going to share my screen at this time and just jot a few reminder notes down into the notebook, the Microsoft notebook that I will be sending your way through Wiley Plus. Okay. So let me just ask in the chat window first. Let me ask in the chat window, are you hearing me okay? Is audio okay? Okay, thanks for the feedback. Sometimes I get comments that make me think that there's a technology issue. So, okay, we'll keep going here. So, all right, so you have your first lecture exam of the semester this coming Tuesday, the 12th. You realize that exam one is on chapters 1, 2, 3, and 4, right? There's 4 chapters of content. Okay, thanks, Christian. It's good to see you back. Chapters 1 through 4. Okay, so on Tuesday, what I need you to do is log in to classmarker.com at 8 a.m. and take that exam, okay? In other words, Tuesday morning, you're logging into the Classmarker website. You are not joining Zoom, right? You're not joining Zoom. There will be no lecture on Tuesday. Okay, so please don't try to join Zoom on Tuesday. Just go right to classmarker.com and take your exam. For your exam, you will have 1 hour and 25 minutes. Okay, so I'm giving you an additional 10 minutes beyond a normal lecture period to take your exam. Okay. For those that are interested, there are going to be 20 multiple choice questions. There will be 20 fill in the blank questions. And then there will be the 4 major essays, which were highlighted on your study guide. Okay. Each of those essays has many questions. Each of those essays has many questions within them. Okay. All right, folks. So is there any, other than the answers to the exam questions, is there anything else you would like to know? Okay, let's do this. Now, I promised you that I would start today's class by giving you an opportunity to ask questions about content. And so let me open the floor to you at this time. We could take maybe, we could take 15, 20 minutes to talk about any content that you're concerned about for the exam. So is there any topics, anything you'd like to ask a question on that I might be able to help you? Go ahead. You can unmute your microphone, type into the chat window, anything you would like to talk about. Okay, folks, now is your opportunity to get a little extra help. So if there's something you're unsure of, now would be the time to ask.
So for like the 4 essays that are going to be on the exam, like, are we writing like full on essay answers, or do you just want like a written response?
Okay, Josie, thanks for the question. Yeah, the kind of essays that you'll get in this course are not the same kind of essay you might answer in an English literature course, right? So in an English literature course, you might be asked to write a paragraph about the protagonist in the English novel. That's not the kind of question you're going to get here. So you're going to get things like label this diagram kind of questions. You're going to get questions such as, put words into this paragraph to make the paragraph make logical sense kinds of questions. So I think that the most appropriate answer to you is just give me the correct response to my question. It doesn't always mean you have to write out full, complete sentences.
Okay, thank you.
Okay, any other questions? Okay, so there's a question in the chat window from Tony. He's asking, how much of the content do you think is the study guide covering for the test? In terms of vocab, Tony, I'm very straightforward. I've only given you hints about 50% of the exam vocab related questions. Okay, so I typically challenge your knowledge using the multiple choice and fill in the blank. So there's going to be 40 of those total, and I've given you strong hints about 20 of them. As far as the essays are concerned, I've told you exactly what the topic is going to be. However, some of the topics are very big, such as, the chemistry topic, where I ask you to know about electronegativity and things like this. The essay from chapter 3, where you have to understand transcription and translation. Those are big topics. So I'm just giving you a hint about what you need to focus on. Okay. So in the chat window, Danielle is asking, can you do an example of calculating the molecular weight of a molecule? Okay. All right. Yeah, we could do that. Okay. Okay, so to calculate the molecular weight of the molecule, obviously there's going to be some information that I must provide to you on the exam. Okay, so please understand, anything you need to calculate with or compute with, I will give you the starting data for. Okay, so I will give you the starting data that you need, and then from that it will be up to you to carry out the process. All right, so let's say, and I'm just going to draw this because I think I can draw it quicker and easier than I can type it out. Let's say you want to know the molecular weight of water, H2O, okay? So what is the molecular weight of H2O, okay? All right, so to calculate molecular weight, to calculate the molecular weight of a molecule, you need the atomic weights, or more appropriately, the atomic masses of each atom. So you need the atomic mass of each type of atom in the molecule. Okay, so I don't expect you to have this memorized, and I also don't expect you to look it up during the exam. But what I will do is I will provide you with this data on the exam. On the exam, I will give you this information, okay? So let's take a look at the type of atoms we have in a water molecule, right? We're going to have hydrogen atoms. I'm going to put a dot there, and we're going to have oxygen atoms as well. Okay, and I can see I'm going to need a bit more space here, so hold on just a second. Okay. All right, so back to this. So I have oxygen atoms and I have hydrogen atoms. Okay, so what we're going to do is write down the atomic mass. Sorry, this is a little crowded here. I didn't manage my space too good. We're going to write down the atomic mass of hydrogen and oxygen. Well, where am I going to get that? Well, right now, I'm just going to look it up on the internet, okay? So I'm going to come over here to good old Google, and I'm going to ask for the periodic table of elements. Okay, and let's see what we can find here. No, that's not useful to me. Let's try this one here. Oh, good grief. Why does everything have to be difficult? There we go. Let me try this one. There we go. Okay, so let me boost up the magnification here. Okay, so from the periodic table of elements, you notice that on this chart, the atomic weight, which is basically the same thing as the atomic mass, is given as an example for sodium. 22.99 is the atomic mass of sodium. Okay, so I'm interested in the atomic mass of hydrogen. That's 1.0078, and I'm getting that from the periodic table. So 1.0078 is the atomic mass of hydrogen. And I need the atomic mass of oxygen, which is from this table here, 15.999. Okay, so 15.999. Okay, so these are the atomic masses of hydrogen and oxygen respectively. Where did I get that? I got that from a periodic table of elements. How will this information be provided on the exam to you? I will simply give you a chart, a table with atomic mass information in it, okay? All right, so we have a column of atomic mass, and now what I'm going to do is just simply write down the number of atoms present in the molecule, number of atoms. Okay, so we see from H2O that in fact there are 2 atoms of hydrogen, and we see from H2O that there is 1 atom of oxygen, right? So what you're going to do is multiply the atomic mass times the number of atoms. So I'm going to put a multiplication in here. All right, and I'm going to bring up my trusty calculator. All right, so for hydrogen, I have 2 atoms of it. Each one of them has an atomic mass of 1.0078. So that's a total of 2. Sorry about that. That went off the screen, didn't it? Come on. Ugh, technology. Do it again. 2 times 1.0078, that's 2.0156. Obviously, 15.999 times 1 is 15.999. And then what you're going to do is add those 2 numbers together. So 2.0156 plus 15.999 is 18. Come on, technology, would you work with me, please? 18.999 0.0146. Okay, that is the molecular weight of water. Okay, so the point of it is that you're starting to get familiar interpreting information from a periodic table of elements. You know, calculating the molecular weight is a very simple thing to do. And you need certain pieces of information, such as the atomic masses of the atoms that are found in the molecule. And those sorts of things will be provided in a data table to you on the exam. Okay, Danielle, does that answer your question for you?
Yes, thank you.
All right. Now, just one final comment. Typically, when you talk about atomic mass, there are units associated with it. You know, this is not a chemistry course, so I'm not going to really be a stickler at this moment for units. But the typical units you would have for atomic mass and therefore for molecular mass would be things that actually is in your textbook called Daltons or grams per mole. Okay, so just have a read over that in your textbook, but it's not a big deal if you don't fully understand it at this time. Okay, other questions? Okay, so if there's no other questions, then I'm going to start some content here today. So one
I
last
have a
time.
quick question.
Okay.
It's not really about content, but like for the lab exam, are you gonna kind of do the same thing and go back and give partial credit if it's not like an exact answer?
Definitely, Sierra. I will be reviewing your lecture exams manually. So, you know, when you finish your lecture exam on Tuesday and the computer gives you an auto-generated score, it's meaningless, right? It doesn't mean anything. So don't take any anxiety or stress from it. Your exam has not been graded until I look at it. So I will be returning partial credit when it can be justified.
Okay, thank you.
Okay. All right. Any final questions? All right, gang. So let's see here. I'm going to get started with new content. Christina, the answer to your question in the chat window. Do we have lab on Tuesday? Yes, we do. Thank you for saying something, Christina. That's something I wanted to mention to you. Let me put it up here. Okay. Yes. So I'm giving you an hour and 25 minutes to take your lecture exam, which means you would finish your exam about 9:25 a.m. and lab is scheduled to start at 9:30. That's too close. I want you to have a break. Okay, so we're going to push off the start time for lab on Tuesday till 9:45. Okay, we'll give you a 20-minute break there. Okay, so lab on Tuesday will start at 9:45 a.m. Okay, so that gives you a 20-minute break between the completion of your lecture exam and the time we need to get back together for lab. Okay. All right. Okay, folks, let's see what we can do here. I'm going to get started with new content, okay? All right. So from last time, we had just started chapter 5. Chapter 5, of course, will not be on Tuesday's exam. So we can just, just pay attention here, take some good notes, but you don't have to worry about this for Tuesday. We were talking about the skin as one of the major components of the integumentary system. We said the skin is basically made of 2 layers of tissue, the epidermis and the dermis, and we started describing what the epidermis is made out of. And so right now I'm defining some of the cells that you find in the epidermis and we're looking at figure 5.2, figure 5.2, which in my book is page 152. Okay. So let me see here. I was talking about melanocytes. That's the last thing. A melanocyte is a type of epithelial cell that makes melanin pigment. And you appreciate that melanin pigment contributes to your skin color. And melanin pigment also does something else that's super mega important. The molecule that is melanin has a unique property. This unique property is it can actually absorb ultraviolet light. I'm talking about sunlight, sunshine. It can absorb it and protect other neighboring molecules that we don't want to absorb the ultraviolet light, such as the DNA of a cell. So what I'm saying to you is melanin is actually protective. When you're out in the sun, it's protective for your cell's DNA, and it helps reduce your risk of developing skin cancer. So melanin has the ability to absorb UV light and protect the DNA inside your skin cells from damage. Okay, so melanin is protecting you from developing skin cancer. Okay. All right, folks. Okay. So the third category of cell from figure 5.2 are these Langerhans cells. Okay, take a look at the Langerhans cells here. Let's see here. Huh, looks like your textbook changed the language on this a little bit. Okay. Yes, panel C in your textbook is called a, well, let me ask you in the chat window. Okay, so figure 5.2, what is item C? Please tell me what item C is. Okay, great. Great. I just want to be sure you're looking at the right things. Okay, so I'm going to use the language of your textbook so I don't confuse you. Okay, so we have dendritic cells. Okay, dendritic cells are also known as Langerhans cells. Dendritic cells are also known as Langerhans cells. Okay. And what's important about these dendritic cells are a type of white blood cell that spends most of its life in the anatomy of the skin, protecting your body from bacteria and other pathogens that might make you sick. Okay, so dendritic cells are defense cells. Dendritic cells are cells of the immune system. They're a type of white blood cell. They're found in the fabric of the anatomy of the skin, and they spend their days trying to protect us from harm from microorganisms that might seek to enter our body and cause problems. Okay. Dendritic cells are also known as Langerhans cells. Okay. All right. So, okay. So much that we could say here. What I'm going to do is define the final fourth cell in figure 5.2, and then I'm going to come back to this topic of immunity in the skin as referenced from the conversation with dendritic cells. So let me complete this list of cells here, and then we'll come back to something that I think you'll find interesting. So the fourth type of cell is a Merkel cell. Thank goodness. Okay, so I'm going to use the, the language of your book. Apparently they relabeled these recently. Item number 4 is the tactile epithelial cell. These tactile epithelial cells are also known as Merkel cells. Okay, they're also known as Merkel cells. What I'm saying is, if you read an alternative textbook, you could be identifying the same cell by an alternative name. Okay, these Merkel cells help give your skin the ability to detect sensations of touch. So these cells help convey a sensation of touch to the central nervous system. Okay. All right, folks. So certainly, you know, are there other kinds of cells in the skin that we're not telling you about? You bet. Definitely. Without a doubt, yes. There are many kinds of cells we find in the skin. Here is just a quick laundry list of 4 typical cells you'll find in the fabric of the skin. Okay. So let's come back here for a moment and talk about immunity in terms of the skin. You know, when you look at a picture, when you look at some of the pictures in your textbook, such as figure 5.1, you see this artist rendering of the anatomy of your skin, and you say, well, okay, that kind of looks interesting, but it's a typical textbook image, you know, blah, 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 boring, 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 who cares, who cares? But remember, folks, your skin is an organ. It is a dynamic living substance that happens to coat the surface of your body. And so think about it. Your outer surface of your body is exposed to the outside world. And so the skin is really the barrier between outside your body and inside your body. So the skin plays a huge role in protecting us from microorganisms in the outside world that might cause us harm if they get into the body. So I'm going to show you a quick little video here. Well, it's not so quick. It's about 7 minutes. But it's a video about immunology in the skin, defense in the skin. As you watch this video, you're not going to understand everything you see. You just won't, and it's okay. Okay, just because you can't understand everything immediately does not mean it's not worth having viewed it in the first place. Okay, so this is a video that's going to introduce some strange ideas to you. But what I really want you to pay attention to is look at the anatomy of the skin. Look at the visualization of the skin and watch how the skin cells move around. Watch how the immune system cells do their thing, okay? So we're not going to get into a deep conversation about immunity right now, but I think this is an excellent video to get you to think about the dynamic nature of your skin and how we take it for granted, even though it's actively protecting us constantly every day. Okay, so here's going to be a video, immunology in the skin. Okay, the video is right, excuse me, it is right here, actually. It's right here. And I'm going to put the link in your notes at this moment, so you can access this again during your private study time. Okay, folks, let's get this rolling. Just watch this and have a look at it. You might see a very strange new world here. Here we go. Okay.
The skin is the body's primary barrier against physical insults and microbial pathogens. It represents a unique environment in which immune cells interact with skin cells to maintain tissue homeostasis and induce immune responses. The skin is composed of epidermis, dermis, and a subcutaneous fatty region. Commensal bacteria, fungi, and viruses living on the skin have beneficial effects in the protection against pathogens and in wound healing. The epidermis is composed of highly specialized epithelial cells known as keratinocytes. They are continuously replenished from just one layer of basal keratinocytes, which divide frequently. Dead cells called corneocytes form the outermost layer and are largely responsible for the barrier function of the skin. In the dermis, cells known as fibroblasts secrete elastin and collagen fibers that form a dense extracellular matrix. Blood capillaries irrigate the dermis, while lymph fluid is drained through lymphatic vessels to lymph nodes, specialized immune structures in which immune cells are activated after pathogen encounter. Diverse and functionally specialized immune cells populate the skin. In the epidermis, a specialized subset of dendritic cells, called Langerhans cells, sample antigen. They project dendrites upward towards the cornified epithelial layer and sample bacterial antigen such as toxins. Langerhans cells appear to be both anti-inflammatory and activatory depending on context. Dendritic cells in the dermis are highly efficient at capturing dead cells and presenting antigen, such as viruses, other intracellular pathogens, or skin-associated self-antigen, to T cells. If dendritic cells are the immune sentinels, T cells are the immune effectors. Healthy skin contains more than twice the number of T cells found in the blood. Most of them are memory T cells that have previously encountered antigen and can be rapidly reactivated. T cells in the epidermis are mostly CD8 T cells, a subset that becomes cytotoxic and kill target cells upon activation. They're long-term residents in the epidermis, mostly disconnected from the circulation. T cells in the dermis are mostly helper CD4 T cells, which have a more modulatory role in the immune response. A variety of other immune cells, such as natural killer cells, eosinophils, and mast cells, are present in the dermis and might be involved in allergic reactions in the skin. Dendritic cells and keratinocytes sense tissue damage, such as wounds or cold sore lesions that occur when latent herpes virus reactivates, and they do that through evolutionarily conserved receptors that recognize pathogen-derived molecular patterns or host-derived molecules that are exposed by cell death, such as DNA. Keratinocytes produce antimicrobial peptides, which can kill bacteria directly, inflammatory mediators, such as interleukin-1 or IL-1, which activate dendritic cells, and chemokines, which recruit neutrophils, macrophages, and T cells. Activated dendritic cells migrate to the lymph nodes, where they present antigen from the site of infection to naive T cells, priming them to activate and differentiate into effector T cells. Activated T cells return to the skin and kill infected keratinocytes to control viral infection or secrete signals that recruit additional immune effector cells. Following viral clearance, memory CD8 T cells persist in the epidermis to provide immunity for future encounters with the same virus. Immune responses can become dysregulated and cause skin disorders such as psoriasis or atopic dermatitis. Psoriasis is a lifelong inflammatory skin disease characterized by scaly reddish plaques. A combination of environmental and genetic factors confers susceptibility to the disease. Physical injury or inflammation can trigger the formation of an acute lesion. The antigenic trigger is unknown, but current models propose that stressed keratinocytes might release self-DNA, which in complex with an antimicrobial peptide activates dermal plasmacytoid dendritic cells to secrete high amounts of the antiviral mediator interferon. Together with pro-inflammatory IL-1 alpha released by stressed keratinocytes, interferon activates dermal dendritic cells to promote T cell differentiation. The earliest recognizable change in the affected skin is the accumulation of T cells and dendritic cells around blood vessels in the dermis. An overt lesion occurs when CD8 T cells, dendritic cells, and neutrophils infiltrate the epidermis. Specialized subsets of T cells secrete soluble mediators like interferon gamma and IL-17, which stimulate the proliferation of keratinocytes, and this produces a marked thickening of the epidermis. Signals from the proliferating keratinocytes act as chemoattractants for infiltrating neutrophils. Crosstalk between immune cells, keratinocytes, and dermal cells thus contributes to tissue remodeling and amplification of this dysregulated immune response. Without treatment, acute psoriatic lesions become chronic lesions. Genetic studies have identified psoriasis-associated susceptibility genes, some of them linking Th17 cells, the subset of T cells that produce IL-17, to psoriasis pathogenesis. Thus, immune cells in the skin exert important roles in maintaining the barrier function against pathogens, but can also become activated by self-antigen or harmless antigen to cause autoimmunity or allergies.
Okay, folks. So there you understand everything you need to know, right? Okay. All right. So let's see here. Let me just ask you in the chat window. Okay. Okay, in the chat window, folks, is the video on immunology in the skin simultaneously cool and crazy complex? I mean, come on, what do you think? Now, hopefully, a little bit of reason that you're in this course is because you're intellectually curious about how your body actually works, because, you know, that's a really good characteristic for a student to have, is some natural curiosity. Hopefully, you know, this helps satisfy some of that. Yes, it is complex. Could we expect you to know everything in the video right now? Of course not, because there was a lot of stuff in there we haven't talked about yet. And honestly, you're not going to get your proper immunology lectures until AMP2, okay? But I wanted to share this with you as a way of giving you a way to visually think about the anatomy of the skin and at least be aware of how involved the anatomy of the skin is in body defense. Do you have any questions you'd like to ask about from having watched the video? Okay. All right. Well, please let me know if you do. We're going to continue our conversation here, folks. We were talking about the epidermis. We talked about cells found in the epidermis. What we're going to do next is move to the layers of the epidermis, which is figure 5.3. Figure 5.3. Okay. So I'm going to mark this as letter C, layers of the epidermis. Okay. When you look at figure 5.3, just take a quick look at figure 5.3 and orient yourself properly along the right-hand side of that figure. Along the right-hand side of the figure is a double arrowed line, a double-headed arrow there that runs vertically top to bottom, and you see superficial on top and deep on the bottom. Okay, so what I'm going to do is actually start talking about the various layers of the skin from the bottom, the deepest layer, and I'm going to progressively work my way up in your notes to the most superficial surface. Okay, all right, so at the bottom layer of your epidermis is a single row of cells, a single row of cuboidal, or in some cases columnar shaped cells, which is called the stratum-based cell. Okay. Stratum-based cell is the deepest layer of cells. It is made of a single layer of stem cells. Okay, it is made of a single layer of stem cells. Okay, so I don't know what you know about stem cells, but obviously stem cells have the ability to build replacement cells for the body. Did you notice in the video that we were watching, there were moments that it looked like a new cell was being made and it pushed all the other cells up towards the surface. So the cell being made was made in the stratum-based cell, and everything sitting above it got pushed further up towards the surface of the body. Okay. All right. So the stratum-based cell is the deepest layer. It's made of a single layer of stem cells, which have the ability to reproduce and create replacement skin cells. Okay. Question in the chat window for you. Question in the chat window, very appropriate for exam 1 coming up. Okay. Here we go. Here it comes. What is the name of the type of cell reproduction the stem cells in the stratum-based cell would perform? What form of cell reproduction will the stem cells perform in the stratum-based cell? What do you think, folks? What form of cell reproduction will these stem cells actually do? Okay, so Sierra has guessed mitosis. Do you agree with her? Okay, all right, well, Sierra is right, and if you agreed with her, you are correct. Okay, so mitosis is going to be the category, the type of cell reproduction used to manufacture replacement cells for the body, right? If we're losing some skin cells off the surface of our body, each day, which we are, we need to replace them. The replacement cells come from the stem cells in the stratum-based cell layer performing mitosis. Okay, let's continue here. All right, so the stratum-based cell is the deepest layer, right? And now we're going to start moving a little bit closer to the surface of our body. The next layer is the stratum spinosum. And you can see that that is labeled along the left side of figure 5.3. The stratum spinosum is typically made of anywhere from 8 to 10 layers of cells, 8 to 10 rows of cells. So if we think about a brick wall, if you could measure out 8 to 10 rows of bricks, that would be basically the composition of the stratum spinosum. Okay, the cells in the stratum spinosum have a very limited reproductive capability. Okay, moving a little bit further towards the surface now, we're moving towards the surface. We're going to have the stratum granulosum. Okay, again, you can see that labeled on the left side of figure 5.3. The stratum granulosum represents a layer of cells which is actually transitioning from being alive to being dead. Okay, it's kind of like watching the old cartoons where you know Bugs Bunny was shot and he goes through a long sequence of dying for the next 30 seconds, right? It takes him forever to pass away. The cells in the stratum granulosum are alive, but they are actively dying, if that sounds, you know, if you can imagine that, okay? So stratum granulosum contains, well, typically 3 to 5 layers of 3 to 5 rows of cells. Typically 3 to 5 rows of cells. The cells in the stratum granulosum, you know, when a cell is pushed up, okay, so when the stratum granulosum makes a new cell, and all the other cells above it start getting pushed further towards the surface. As a cell transitions from the stratum spinosum layer to the stratum granulosum layer, it is alive. It is alive, but the longer it spends in the stratum granulosum, the closer it is to dying. Okay, so the stratum granulosum is where cells go to die. Okay, new cells that enter this layer are alive, but then gradually begin to die. Okay, and that cell that death process is intentional. Okay, there is a purposeful intended cell death pathway called apoptosis. So cells in this layer are executing a type of genetically defined cell death known as apoptosis. Okay. All right. Okay. So let's see. Let's see if we can. Let's see if we can ask someone in class about this. Okay, hold on a second. Let me get my roster out here. Okay. How about Rebecca Panabaker? Rebecca, could you unmute and say hi to us, please?
Hello.
Hi, Rebecca. How are you this morning?
Good. How are you?
Okay, thank you. Hey, Rebecca, why is it the case that as cells migrate towards the surface of the body, that they start to die? Why is that?
Is it because they're getting closer to the air?
Huh, that's an interesting idea. Well, so are you telling me that air is somehow toxic to cells?
Yes.
Okay, that's not correct, Rebecca, but it was a good shot. Would you like to change your answer?
I honestly couldn't tell you the correct answer.
Okay, thanks, Rebecca. I appreciate your participation today. So folks, here's what I'm fishing for. As cells migrate, as they travel closer to the surface of the body, they are traveling further away from their blood supply. They're traveling further away from their blood supply. So let me ask you this in the chat window. Hold on a sec. Here we go. Does epithelial tissue have blood vessels? Does epithelial tissue have blood vessels? Do you remember this conversation we had in chapter 4? Okay. Okay, it does not. Epithelial tissue does not have blood vessels. Well, what's the epidermis, right? Isn't the epidermis simply the specialized epithelium of the skin? So the cells in the epidermis, which are really simply epithelial cells of epithelial tissue, as they get further towards the surface, they are actually walking away from their source of life, which is the bloodstream. So as cells migrate up towards the surface, as they finally reach the stratum granulosum, they are at this point getting so far away from their blood supply that they don't have enough access to nutrients or oxygen in the blood supply to maintain their life. And that triggers this process that we are defining as apoptosis. Okay. All right. So let's put that down in your notes. Okay. This process is triggered as cells travel further away from their blood supply, meaning that the cells no longer have abundant access to oxygen and nutrients. Okay. Folks, let me ask you in the chat window. Does the answer to why cells are dying in this context make sense to you? I mean, if you walked away from your refrigerator and you had no other source of food, right, you would starve to death. That's basically what's happening here. If you somehow walked away from your source of oxygen, your source of air, you wouldn't last very long, would you? These cells and these tissues, they need access to oxygen and nutrients, but there's no blood vessels running through the epithelium. And so the further they migrate away from the blood supply in the dermal layer of the skin, more likely they are to die. It seems to be the case that when cells reach this particular layer that we have named stratum granulosum, the lack of oxygen is just too much and the cells start to break down. Okay, let's continue here. All right, next up, we have the stratum lucidum. So we're getting even closer to the surface at this point. The stratum lucidum is a fourth layer of the epidermis that is not found in all sections of the skin, okay? This layer is not found in all sections of the skin around your entire body. Okay, so we don't find it everywhere. So what's the deal here? The stratum lucidum is an additional layer of epidermis that is found in areas of your skin that is typically being abused by physical abrasion. So think about it. You know, a lot of times folks, you know, if you were to take your socks and shoes off, you would see, you know, the bottom of your foot and you would see the calluses on the bottom of your foot, right? And a lot of times if you walk around barefoot, particularly in the summertime, those calluses get even thicker, don't they? Because you're really, you know, you're really stressing the underside of your foot there. So any place where you're grabbing things with your body's anatomy, with the skin, such as your fingertips, you know, particularly if you're very much involved in a manual labor profession, which you know, which starts to damage the skin, you're going to build up calluses. So think of it this way. Stratum lucidum is typically any place on the skin where you might typically build a callus because you're doing some sort of manual interaction with the skin, you know, using your hands to build things, walking barefoot, that type of thing. You will not find stratum lucidum, okay? You will not find stratum lucidum on the skin that's part of your forehead. Why not? Because you're typically not rubbing your forehead against the wall. You're typically not picking up a cup of coffee with the skin on your forehead, right? You're not interacting with harsh objects with the skin on your forehead. So any skin that's not interacting with things on a daily basis, not being exposed to friction, you're not going to have stratum lucidum in those locations. So this is only found in areas of the skin that are exposed to abrasion and friction, such as the soles of your feet, palms of your hands and fingertips. Okay, for these locations, you can find calluses. Part of the callusing process is building this additional layer of the epidermis known as the stratum lucidum. Okay. Cells in the stratum lucidum are completely dead. They are metabolically inactive. All right, finally we get to the most superficial layer. In the video, these most superficial cells were called corneocytes, right? You remember corneocytes? You can see in figure 5.3 along the left side that the most superficial layer is the stratum corneum. Stratum corneum. Okay, so maybe I should remind you here that we're going from We're going from deep to superficial here in our notes, deep to superficial. Okay. Stratum corneum. Okay. The stratum corneum is the most superficial layer of skin cells, typically made on average 25, 30, 35 rows of cells. Okay. Typically formed from 25 to 35 rows of dead cells. Once again, these are even though we're calling this layer stratum corneum, the stratum corneum is made of dead keratinocytes. I mean, that's the majority type of cell you're going to find here is dead keratinocytes. It may be of interest to you to know that typically these cells of the stratum corneum are sloughed off the surface of the body millions of times a day. Okay, so let me ask you this question in the chat window. Do you have an allergy to dander, or do you know anybody that has an allergy to dander? Okay, dander is basically sloughed off dead skin cells, and most times, folks might have an allergy to the dander of an animal or a pet at home, such as a cat or dog, or your pet chimpanzee, or whatever you folks have at the house. Okay, and so allergies can be produced. Allergies can develop from a sensitivity to these dead skin cells that are floating in the air around us. Okay. So let's see here. Stratum corneum, these cells slough off the surface of the body millions of times a day. Dead skin cells, dander in the air around us triggers allergies in some persons. Okay. Now, if you think for just a moment, there really is a logic to why Mother Nature is shedding these dead skin cells. Here's the basic, well, here's one piece of logic that scientists can say is true. Okay, so let me just do a little bit of drawing here. Okay, let's say that this is the surface of the skin on your arm. Obviously, it doesn't have to be the arm. It could be your leg. It could be your head. Who knows? Could be any part. But what I want to point out is that the topmost layer of cells, which you now know are corneocytes, which are also just the same thing as dead keratinocytes. Okay, so they're going to be lining the surface. So I'm just going to draw an overly simplified diagram where you have some of these dead skin cells on the surface. I'm going to draw a really large one here, simply to give you some visualization. Okay, so these are the dead corneocytes. Okay, the point that I'm trying to make here is that every day the surface of our body is assaulted with microbial invasions. Okay, there are microorganisms floating in the air around us. You cannot avoid it, folks, right? Our air is loaded with life. It's absolutely loaded with life. And so there are microorganisms floating in the air of the room you're sitting in right now. You just can't see it. And the point of it is that you might have a bacterial cell that was floating in the air sit down on your cell. Or you might actually have a virus particle sit down on your skin cell as well. Well, what these microorganisms tend to want to do is find a food source, and your body could conceivably be a very nice food source, okay? And so these microorganisms might want to cause us harm. Well, honestly, the honest answer is these microorganisms want to live. They want to live. And if in consuming parts of your body to allow them to live, it makes you sick, well, that's what we call disease. That's what we call infection. Okay, so the purpose of it here is that Mother Nature has designed a system where the outer surface of our skin, which is continually being assaulted, will respond to that assault by having the skin cells literally detach from the surface of your skin and float away. And obviously, if your skin cell floats away and there's an attached virus particle with it, that virus particle also floats away. So the skin cell and potential pathogen detach from our body. Okay, so this is actually a defense mechanism, a protective mechanism to have our body made of cells that actually slough off millions of times a day, because in doing that, we also detach microorganisms from the surface of our body that might seek to get into our body and ultimately cause us to get sick in some way. Okay. So, you know, while that is a protective mechanism for us, we also have to admit at the same time that if you're sitting in a room full of people, if you're sitting in a crowded bus, if you're sitting in a crowded train, if you're sitting in a crowded airline cabin, an aircraft cabin, that when somebody detaches one of their skin cells with a pathogen attached to it, well, that thing is floating in the air, and in the next moment of time, you unfortunately, someone else sitting next to you could inhale it into their nostril, and all of a sudden it's down into their respiratory tract, right? So it's kind of a give and take scenario there, which is one of the reasons, particularly right now when we're dealing with the coronavirus pandemic, that wearing a protective layering, I'm talking about a mask over your nose and mouth, is actually helpful to block the entry of these microbes into our body. Okay, folks, do you have any thoughts or questions right now? Okay, so you know, if you know anybody that has dandruff, I mean, in various moments of my life, I've had dandruff and I've had to get some medicated shampoos to deal with it, right? Dandruff is simply an excessive shedding of keratinocytes from your scalp, right? There's nothing to be ashamed of. It just is one of those things. Okay. All right. Any questions? Okay, folks, so that's a basic coverage of the epidermis. Number 3, the epidermis. Now I'm going to switch gears and start talking to you about, the dermis. Okay, let's start talking about the dermis. Once again, if you're not clear what I'm referring to, just take a look at figure 5.1 and you'll see the terms epidermis and dermis are along the right side of that figure. In fact, I'm going to reference that figure again here in your notes so that you know to look at it. Okay. Okay, so the dermis, remember, is the connective tissue. The dermis is made of dense, irregular connective tissue. This layer of the skin is made of dense, irregular connective tissue. Okay, so that's a chapter 4 topic, right? When you come across dense irregular connective tissue in your study time, you should immediately be thinking, oh, dermis, dermis, dermis. Okay, so dense irregular connective tissue is the type of tissue that makes up the dermis, and it contains a great deal of collagen and elastic fibers. Okay, question in the chat window, question in the chat window. Here it comes. If the dermis contains an abundance of collagen fibers, what type of cell do you expect to find here? What type of cell do you expect to find? Okay, so key to this is the word collagen. Collagen. What kind of cell do you expect to find here? Well, Rebecca, you might find some reticular cells, but that's not what I'm fishing for. I'm fishing for the cell which is known as the collagen factory. The collagen factory. Any guesses here? Any additional guesses? Okay, let me help you out. The answer begins with the letter F as in Frank. F as in Frank. Okay, thank you, Sierra. Absolutely, fibroblasts, right? You know, many cells of the body have the capability to make collagen, but fibroblasts are famous for the quantity and large volumes of collagen that they manufacture and secrete into the extracellular matrix, right, of connective tissue. Okay, so this dermis, because it is a connective tissue, has relatively few cells, but those that are present include fibroblasts, macrophages, and adipocytes. Okay, fibroblasts, macrophages, and adipocytes. Hopefully some of those cells are coming back to you from our discussion in chapter 4. Okay, if you look very carefully at the picture of figure 5.1, notice the presence of the following structures. Notice the presence of blood vessels. Notice the presence of glands. Okay, do you see there along the left-hand side? If I go down figure 5.1 and just scan the left side of terms, notice I see a sebaceous gland there. Okay, that's an oil gland. If I continue further down, you see the eccrine sweat glands and the apocrine sweat glands, right? Okay, so you're going to find glands embedded in the dermis. You're going to find hair follicles as well. Okay, so the analogy I like to use here, and I might have used this previously for another thing, but it's still legitimate here. When you think about, when you think about the dermis in terms of what its composition is similar to Jello that has fruit embedded in it, right? Have you ever eaten a fruit cup of Jello where there's fruit actually in the Jello itself? Like some pears or peaches in your strawberry Jello. Okay, so the anatomy of the dermis is similar to Jello with fruit embedded in it. Okay. Obviously, the fruit are all these structures I'm talking about: blood vessels, glands, hair follicles, that sort of thing. And then everything else embedding it in there would be the extracellular matrix. That would be the jello. Okay. All right, folks, I want to turn your attention to two regions of the dermis, okay? As you're looking on figure 5.1, look at the bracket of the dermis on the right-hand side. Take a look at those two regions called the papillary dermis and the reticular dermis. The papillary dermis and the reticular dermis. So the dermis can be subdivided into two regions. We'll just write these out here. First, we have the papillary region. Okay, and let me just say a couple things about the papillary region. Okay, in the papillary region, do you notice a structure along the left-hand side called dermal papillae? Notice the dermal papillae. Okay, notice the dermal papillae. Okay, the dermal papillae are kind of like finger-like projections that project upward into the bottom surface of the epidermis. The purpose of the dermal papillae is multiple. There are multiple things the dermal papillae do for the dermis. Okay, so let's list these out here. So the dermal papillae increases the available surface area to lock in with the anatomy of the epidermis. In other words, that provides good structural integrity. I don't know if you can see my fingers locking together in the video window, but what I'm trying to suggest is bring your hands together. Bottom hand is the dermal papillae. Top hand would be the epidermal pegs. You see the epidermal pegs on the left side there? They're locking together like a tight fitting jigsaw puzzle. That provides good structural integrity to bond the epidermis to the dermis. So the dermal papillae are used to bond, or you might say stitch, the dermis to the epidermis, right? So this increases the structural integrity of the skin. I will point out here as well that if you look very carefully in figure 5.1, look very carefully, you will notice some red and blue tiny little loops, that are embedded up into the dermal papillae. These are capillary loops. So what I'm trying to tell you folks is the dermal papillae represents that region of anatomy in your skin where you get the blood supply that is closest to the surface. Okay, your blood supply in terms of your blood vessels never gets any closer to the surface than the dermal papillae. So the dermal papillae are used to bring capillary vessels, right? So capillaries are the tiniest blood vessels of the body. To its most superficial point, right? You're never going to get a capillary loop normally that extends beyond that. So this is the most superficial region of your skin that the blood supply is stored in, okay? So they're used to bring capillary vessels and blood to its closest superficial point in the body. Okay, it is also true, let's see here. If you look carefully on the right side of figure 5.1, do you notice the phrase free nerve ending? Free nerve ending. Free nerve ending You notice that it is also inside a dermal papillus. In other words, not only do we bring blood vessels up into the dermal papillae, we also bring nerve endings into the dermal papillae. And while I won't go into the details right now, some of those nerve endings endow our bodies with the capability to receive sensations that are communicated into the central nervous system so that we can receive sensations of warmth or coolness or tickling or itching or even pain if the skin is being inappropriately stretched. These nerve endings provide information that leads to the sensations of warmth, coolness, pain, tickling, and itching. Okay, so there's different kinds of nerve endings that are embedded in the dermal papillae, and that will be a talking point for our future conversation. Okay. All right. So let me also mention to you the reticular region. I don't have as much to say about that, but we'll put it in your notes. Let's see. Oh, shoot, I'm out of time, aren't I? Okay. All right. Let me just write reticular region down and I will stop. Okay. Sorry about that, folks. Okay. So we'll stop here and pick up this time next Thursday. So folks, let me remind you, Tuesday, log in the class marker at 8 a.m., take your exam. Please come join me for lab at 9:45 on Tuesday. Folks, have a good weekend, study really hard, okay? Give yourself breaks, but study as hard as you can, and good luck on your exam, okay? See you later.
Thank you.